Hello everyone. In this lessons learned video, we're going to discuss the basal ganglia. The basal ganglia are sometimes called the basal nuclei or the cerebral nuclei. They're a group of gray matter structures in the subcortical regions that are going to be involved in the activation and inhibition of motor commands. They're mostly going to communicate through unmyelinated fibers, and they're mostly going to be nuclei, meaning that they're collections of nerve cell bodies. So they're going to be gray matter, or seen as gray matter. So the name basal ganglia or basal nuclei implies that there's going to be multiple nuclei in there, right? So one of the major ones is the striatum or the CPU, which you can see over here. Now, the striatum is made up of two nuclei. One of them is the caudate, which you can see is this red structure that goes sort of around. I've redrawn it over here, and you can see it has a head, a body, and a tail. So that's the caudate nucleus. It's going to be the wall of the lateral ventricles, the lateral wall of the lateral ventricles. The putamen is slightly more lateral to the caudate nucleus, right? And it overlaps from this lateral view a little bit on its head. So it's this structure you can see right there. Now, at the tail end of the caudate, we're going to have the amygdala, just for reference. It's not part of the basal nuclei. The globus pallidus is over here. It's actually going to be medial to the putamen. So if I had just colored in the putamen, you wouldn't be able to see this, right? So it's like a little bit more into the page. And the globus pallidus has two functionally distinct units. One of them is the globus pallidus internal, the internal globus pallidus, and the external globus pallidus. Now, we also have the subthalamic nucleus, which is above the tail of the caudate, but below all this stuff on top. Should also mention that the thalamus is behind everything in this picture, right? So it's most medial to the putamen, the caudate, and the globus pallidus. The substantia nigra is going to be this nucleus within the midbrain, and it's going to look dark on brain sections because a component of it, called the substantia nigra pars compacta, is going to be dopaminergics, and the metabolic byproduct of dopamine, or one of its metabolic byproducts, is a substance called neuromelanin, and it looks dark. And so that part of the substantia nigra that makes dopamine is the substantia nigra pars compacta, but we also have the pars reticulata, which makes GABA. So those are going to contain GABAergic neurons. So just show you another view of the basal nuclei. I've recolored some of the stuff. The putamen and the caudate are the same color here because they're collectively known as the striatum. And so you can see that the putamen is right here, but over here we're kind of cutting across it, right? So we're kind of going like this across the putamen. Same thing with the caudate. Even though the caudate has this this sort of shape, right? Over here you can only see the head of it, or maybe actually more the body of it, right there. Now, I said that the globus pallidus has two distinct segments. There's going to be the external, right? So this is going to be kind of more lateral, this blue one. And then there's going to be an internal segment that is more medial, right? It's kind of further into the page on this lateral view. The thalamus is going to be medial to all these nuclei, so it's going to be over here. And then the subthalamic nucleus is going to be under all of this, so right there. And then the substantia nigra is going to be in the midbrain. That's further down, right? So hopefully that gives you a better view of the anatomy. So now let's talk about the circuitry here of the basal nuclei. So what you see on the right side is a simplified diagram of what you see on the left side. So let's start from the bottom. We see that we have the substantia nigra, but we said that there's the pars compacta and the pars reticulata. So the pars compacta is shown right here with the C, and then the reticulata is shown with the R. Okay, S and R is pars reticulata. The subthalamic nucleus is right here, as it is over here on the coronal view. The globus pallidus internal is right there. The external globus pallidus is this blue one. And then I've grouped together the caudate and the putamen, right? Collectively known as the caudate putamen complex, or just the striatum. Then the thalamus is kind of more medial and up here, right? Then we have the cortex. Okay, the cortex is just like the region over here. So these basal nuclei in general are going to communicate with each other and then send an output to the thalamus. And then the thalamus is going to influence the activity of the motor cortex. So before we talk about this, I want to go over an important principle, because if I don't, we'll make the rest of this very complicated. Let's say that you have a nucleus here, right? It can be any of the nuclei above, right? It can be any of these, right? Just take it as a generic nucleus. And let's say we have A, B, and C, and D. Now, if I go ahead and I excite B, but let's say that B sends inhibitory projections to C. Okay? And then C sends also inhibitory projections to D. Now, notice what would happen if, say we have another nucleus as the start. That's going to go ahead and excite A, right? If we excite A with an excitatory projection, that means that this is excited, right? Or we can just say up arrow. Then this is going to have more activity, so it's going to send more excitatory input to B. So B is also excited. If B is more excited, then that's going to send more inhibitory projections to C. So C is now inhibited. And if C is inhibited, it's not going to send as much inhibitory output to D. So D is what we call disinhibited. What that means is that we're taking away inhibition, right? Which is effectively almost the same thing as excitation. Think of it as two negative signs being multiplied, right? If C usually inhibits D, but you prevent the inhibition, you in effect excite D. Okay, so in these diagrams and in these circuits, whenever you see two inhibitory projections coming into the same nucleus and going out from the same nucleus, which is C, the last nucleus, which is D, is going to be disinhibited, right? This is going to cause disinhibition. And this disinhibition can be treated as almost the same thing as excitation, even though it's not exactly the same thing. So the activity of D is going to increase because we're taking away the normal inhibition that would usually occur. So with that in mind, let's talk about the two pathways of the basal nucleus. So this direct pathway is going to facilitate movement and help initiate voluntary movements. So we said that excitation was, okay. All right. So it's going to begin by the cortex and the substantia nigra pars compacta exciting the striatum. And more importantly, this is going to happen through D1R receptors from the striatum. What this means is the substantia nigra is always going to be sending dopamine to the striatum, which is, we can say dopamine is DA. But in this case, the striatum, it's sending the dopamine to dopamine 1 receptors on the striatum. And dopamine 1 receptors, when dopamine binds to them, they make the cell activated, right? So that's why the signal is excitatory. Now, the striatum is going to be inhibiting the internal globus pallidus. All right. And then the globus pallidus is usually going to be inhibiting the thalamus so that we don't have movements at rest. But here, notice what happens. We have an inhibitory signal coming into this nucleus, the globus pallidus internal, and an inhibitory signal going out, which means that the thalamus, right, is going to be disinhibited, meaning its activity is going to increase. Now, what does the thalamus usually do? The thalamus sends excitatory output to the cortex, right? So if we increase the activity of the thalamus, it's going to send more excitatory output to the cortex, and the cortex, when it's activated, is going to lead to motor output. So we're going to have more motor output, and we're going to initiate that movement as we're supposed to in the direct pathway. So that's how the direct pathway works. And if you want to follow the trace of the entire activity here, the activity in the striatum increases, which leads to more inhibitory output to the GPI. So the globus pallidus internal has less activity, so it's not going to be inhibiting the thalamus as much. So the thalamus is then disinhibited. Sorry. The indirect pathway is the other pathway. It is going to be involved in terminating motor commands, meaning stopping movement. So the way this works is that the cortex still sends excitatory input to the striatum. But in this case, we're going to have the substantia nigra pars compacta sending dopamine again to the striatum, but this time it's sending it to dopamine 2 receptors as opposed to dopamine 1 receptors. Now, when you send dopamine to a dopamine 2 receptor, it's going to cause an inhibition of the cell that's receiving dopamine. So the striatum is going to be inhibited, even though the substantia nigra is still sending the same molecule, dopamine. Then this striatum is going to then inhibit the external globus pallidus. Now, the external globus pallidus, is usually going to be inhibiting the subthalamic nucleus. So far, let's just see what has happened, right? We sent inhibitory input and excitatory input into the striatum. So you might wonder what happens to the striatum in this case. Well, in this case, it's actually going to have a net excitation because the cortex is more powerful than the substantia nigra pars compacta, which is this little nucleus, but the cortex is pretty large. So it's going to cause a net excitation of the striatum. Then this means that the striatum sends more inhibitory output to the GPE, right, to the external globus pallidus. So the external globus pallidus, its activity is going to decrease. But notice again, we have 2 inhibitory signals going in and out of a nucleus. So this end nucleus, the subthalamic nucleus, is going to be disinhibited, right? So its activity is going to increase. The subthalamic nucleus usually is going to be exciting the internal globus pallidus and the substantia nigra pars reticulara, which I don't know where the R went, but it should be this. Okay. Now these two nuclei, since we're exciting this subthalamic nucleus, it's going to send more excitatory output to the substantia nigra pars reticulata and the internal globus pallidus. So these two nuclei are going to have increased activity. Now, these two nuclei are usually going to be inhibiting the thalamus, right? Just like the other pathway, the globus pallidus internal is inhibiting the thalamus, and the substantia nigra pars reticulara is also inhibiting the thalamus. Now, if we increase the activity of each nucleus, which we have, right, these two have more activity, and if they inhibit the thalamus, we're going to inhibit the thalamus, in effect. So this is going to be having lower activity. And the thalamus usually excites the cortex at rest. But in this case, if the thalamus is being inhibited, it's not going to be able to do this. So the cortex is going to be inhibited compared to usual, which means that we're, in effect, not going to get this motor output. So we're going to stop movement. Now, you might wonder what happens when we have pathology of this circuit. All right. And we're going to have different pathologies called dyskinesias. So we can have what is called let me do it in white, hyperkinetic dyskinesia, which means that we have excessive movements. This would be something like hemibolismus, which is one condition. Or we could have a hypokinetic dyskinesia, where we have not enough movement. The patient cannot initiate movements. And so this is not enough movement, you could say. And an example of this can be Parkinson's disease. Another example of a hyperkinetic dyskinesia is going to be Huntington's chorea. And Huntington's chorea is a genetic disorder. Chorea means to dance, so that it causes dance-like movement of the limbs. So let's talk about some of these conditions. So let's start with Parkinson's disease. You may have looked at this circuit and said, okay, look, the cortex is sending excitatory input to the striatum. The substantia nigra pars compacta is sending inhibitory input into the striatum, but we're getting a net excitation of the striatum. So does that mean that this substantia nigra pars compacta is basically useless? Well, it's really not, because in Parkinson's, this substantia nigra pars compacta neurons are selectively degenerated. Okay, so imagine that the person has Parkinson's, right, and that this just goes away. Well, you're not going to have this inhibitory input into the into the striatum. So imagine that this goes away. If you take away the inhibition, you're in effect going to have excitation, right? So it's going to excite this pathway even more than usual, right? So we can represent that with a bigger arrow. And then if you follow the circuit again, if this is really excited more than it should be, then we're going to have a more strong inhibition of the globus pallidus external region. So this is represented by bigger inhibitory arrow. And that means that we're going to disinhibit the subthalamic nucleus a lot more than we usually should, right? Which means we're exciting the substantia nigra pars compacta and the globus pallidus internal a lot more. Which means the thalamus here, right, because these two send inhibitory outputs to the thalamus, is really inhibited. Which means the cortex is also really inhibited, meaning we really have a difficult time generating motor output. So people with Parkinson's have the will to move, but not the ability to. It's difficult for them to initiate movements. So that's how Parkinson's disease happens. So now, let's consider a different condition. Let's consider Huntington's chorea. So let me delete all the arrows that we've drawn. The new ones, at least. Alright. Huntington's chorea involves a selective degeneration of the striatal output neurons, so these striatum CPU neurons, the caudate and the putamen. If you degenerate these output neurons, you're not going to get as much inhibitory output to the globus pallidus external. So if you don't have inhibitory output, you in effect have disinhibition, right? You're taking away inhibition. So that's actually going to cause disinhibition, which, as we said, is similar to excitation. So it's not going to be inhibited anymore. So if you follow this whole circuit down, which we'll do right now, if the globus pallidus external has more activity in it, it's going to then inhibit the subthalamic nucleus more, which means that the subthalamic nucleus should be inhibited. And less activity in the subthalamic nucleus due to this inhibition is going to cause less excitatory output to the globus pallidus internal and less excitatory output to the substantia nigra pars reticularis. So these are going to have less activity. And if those are going to be inhibited, they're not going to be sending as much inhibitory output to the thalamus, right? So they're not going to be doing this as much, which means the thalamus, in effect, is going to be excited because you're taking away the inhibition from it. Then the thalamus is excited in the indirect pathway, so it's going to be exciting the cortex, so the cortex will actually have more activity as opposed to less, which means that we actually do get motor output here. So you can see that we have motor output in the indirect pathway where we're trying to inhibit motor output. So this leads to involuntary movements at rest. And specifically for Huntington's chorea, it's going to be a dance-like movement of the limbs. Now that's Huntington's chorea. Now let's talk about hemibolismus. Alright. So hemibolismus is another hyperkinetic disorder. It's going to be caused by a small stroke in the subthalamic nucleus. So this subthalamic nucleus is going to be lesioned. So if you don't have this subthalamic nucleus or it's lesioned, you're going to have less excitatory output to the GPE and to the SNR. Okay. What that means is that the activity of these two nuclei is going to decrease, which then would mean that if their activity decreases and they're supposed to be inhibiting the thalamus, they're no longer inhibiting the thalamus, so the thalamus is in effect going to be released from that inhibition and going to be excited or disinhibited. And a disinhibition of the thalamus would cause more activity in the thalamus, which means we're exciting the cortex more. So this becomes an up arrow. And then we are no longer inhibiting motor output within the indirect pathway. So we're getting increased motor output. And once again, this causes hemibolismus, which is ballistic movement of the limbs due to this small stroke in the subthalamic nucleus. So I hope that helped you understand the anatomy and the physiology of the basal ganglia. Again, I would go through these pathways, explain to yourself if you don't yet fully understand it, and also go over the anatomy here. Yeah. Hope it helped. See you in another video.